Hello, everyone, and welcome to today's webinar, Unveiling Insights, Leveraging Single Cells, Transcriptomics, and Epigenomics for Biomarker Discovery. I'm Cassie Saltman of LabRoots, and I'll be your moderator for today's event. Today's educational web seminar is presented by LabRoots and brought to you by Takara Bio. To learn more, visit takarabio.com. We encourage you to participate today by submitting any questions you may have during the presentation. To do so, simply type them into the Ask a Question box and click Send. We'll answer as many questions as we have time for at the end of this presentation. You may also submit any technical issues here as well if you have trouble seeing or hearing the presentation. I'd like to now welcome our speakers, Dr. Derek Janssens, Hartwell Postdoctoral Fellow, Basic Sciences Division, Fred Hutchinson Cancer Center, Lab of Dr. Stephen Henikoff, and Dr. Ilaria Iacobucci, Principal Scientist, St. Jude Children's Research Hospital. Dr. Janssens, you may now begin your presentation.
Hello, thank you all for tuning in. I'm excited to present as part of this LabRoots seminar with Takara. My name is Derek Janssens, and I am a postdoc in Stephen Henikoff's lab at the Fred Hutchinson Cancer Center. And today I'm excited to tell you about how we've been using the iCell8 platform to perform scalable single-cell profiling of chromatin modifications with CUT&Tag. And in the Henikoff lab, we're broadly interested in genome regulation and the role of chromatin in that process. And chromatin modifying enzymes play a critical role to partition the genome into both transcriptionally or developmentally repressed chromatin domains, as well as transcriptionally active chromatin regions. And these chromatin modifying complexes can be broadly divided into two groups known as polycomb group proteins, which are critical for maintaining developmentally repressed genes, and do this in part by catalyzing the modification to the histone H3 protein, which is part of these nucleosome complexes that package the genome, where it catalyzes histone H3 lysine 27 trimethylation. So that mark is located on regions that are developmentally repressed. Now, in contrast, the trithorax group proteins maintain developmentally active regions of the genome and also modify the histone H3 protein on lysine 4 and catalyze methylation. And these complexes play critical roles in regulating developmental transitions in our cells, but studying the role of these complexes in heterogeneous tissues in which these developmental transitions occur in relatively rare populations of cells has been extremely challenging, in part because methods to examine the activity of these complexes at a single cell resolution have been lacking. And so to address this issue, several years ago, a postdoc named Hatice Kaya Akor, along with Steve, developed this method called CUT&Tag. And I'll go through this briefly, but basically the way it works is the cells of interest are bound to concanavalin A-coated magnetic beads for separation during wash steps. And these are then permeabilized to allow an antibody that targets a chromatin-associated protein of interest to freely diffuse into the nucleus and bind to its target. This antibody is then followed up by what's really the key ingredient of CUT&Tag, and that's this protein A TN5 fusion. And you may recognize the TN5 cut-and transposase enzyme because it's also utilized in the popular ATAC-seq method. But the difference is here, this TN5 is tethered in place to the antibody by this protein A, such that upon activation with magnesium, TN5 will drop its payload of these adapters locally, directly adjacent to this chromatin-targeted, chromatin-associated protein. And these adapters and tagmented DNA can then be extracted and is ready for amplification to generate a genome-wide sequencing library to basically map the footprint of the locations where this antibody-targeted protein had been associated across the genome. And one of the advantages or really powerful aspects of this method is that this tagmented chromatin remains intact within the cells up until library preparation. And what's so critical about that is it makes the cut-and-tag method compatible with a number of single-cell genomics platforms, such that the antibody binding, TN5 binding, and tagmentation are carried out in bulk, and these cells can then be arrayed for generation of single-cell libraries either in a nanowell platform, such as the Takara iCell 8, or in the popular droplet-based method like 10x Chromium. And so initially we were exploring both of these platforms and noticed some key differences. So whereas in these nanowells, we were able to capture a larger library per cell, in each experiment, the number of cells captured was relatively limited to 1,000 to 2,000 cells per experiment. Now, in contrast, these droplet-based methods usually produce a smaller library per cell, but you could capture many more cells. And so part of the reason why we were capturing more unique reads per cell is that the actual reaction chemistry could be modified by the user in these nanowells. And so we went on to further optimize this reaction chemistry by basically treating the tagmented chromatin with a low concentration of SDS and then heating it to 58 degrees. And this essentially loosens the chromatin up, bumps off the TN5, and prepares the tagmented DNA for PCR amplification. So this SDS is then crunched by Triton, and we performed a titration experiment on the iCell 8 where we were able to identify the optimum SDS and Triton concentration to capture the maximum unique reads per cell and increase the library size of individual cells. And so the next challenge that we wanted to overcome, now that we had this optimum chemistry in place, is to increase the number of cells that we could profile in a single experiment. And to do this, we turned to a strategy called combinatorial indexing, which I'll describe next. So in this workflow, and for the remainder of this talk, I'll describe a flagship experiment where we applied this to profile peripheral blood mononuclear cells, in which we mix these PBMCs from two different donors. I'll explain why that's important in just a moment. But basically, we again mix our cells and bind them to, in this case, WGA-coated beads, which again allow magnetic separation during washes. And we then carry out label the chromatin for either these active modification to the histone H3 lysine 4 residue, or the developmentally repressive modification, histone H3 lysine 27 trimethylation. And this is done in bulk in parallel tubes. And following antibody labeling, the next step is that the sample is then arrayed across a 96-well plate. And this allows the first round of cellular indexing, in which differentially barcoded PATN5 complexes are added to each of these individual wells, and the chromatin is then tagmented. And following this first round of indexing, in which now the cells all carry one of 96 different barcodes, they're then mixed back together in a split pooling reaction, and then again distributed out on the iCell 8 across these 5,184 nanowells for a second round of cellular indexing during the PCR step. And the power of this method is that basically by limiting the number of cells dispensed into each one of these second nanowells to between 10 and 20 cells, we can limit the chance that any one of the given cells that falls in one of these nanowells arose from the same well of the 96-well plate. And so without ever having to isolate individual cells, we can still generate single cell libraries based on their differential indexes. And so to just give you an idea of what this looks like, whereas previously, we were trying to identify wells with a single cell, as shown here, in this modified sky cut-and-tag approach, we just dilute the sample to between 10 to 20 nuclei per dispense, and then use the entire plate, because now each well has, a Poisson distribution centered around 10 to 20 nuclei. And so we no longer need to create a filter file, but rather dispense across the entire plate for the subsequent DCR reactions. And this generates a robust library shown here, where that includes the single cell libraries from all of the cells from this one of these experiments, which is this 21.8 nanomolar library. And here I'm just showing you a schematic of what the actual content of one of these reads includes, where you see there's two different barcodes, which are read out by one read on a next generation sequencing instrument. Then there's a second read to do one side of the genomic DNA. A third read captures the opposite side of the genomic DNA, and the fourth read does the second, or the third and fourth index. And if we then look at this data, you can see that both the repressive and active marked cells produce this stereotypical nucleosomal ladder, which is characteristic of the cut-and-tag data, where we see both subnucleosomal, mononucleosomal, and dinucleosomal fragments, which is just characteristic of the TN5 tagmenting around either a single nucleosome or two nucleosomes. So that's a good sanity check and suggests that our library is indeed, high quality. And the next we just looked at the distribution of the number of unique reads per cell, shown here, with unique reads on the x-axis and the number of cells falling in each one of these bins on the y-axis. And you can see that for the repressive chromatin modification, we're capturing a slightly higher, on average, a slightly higher library per cell. And we then just set this lower threshold at around 500 reads per cell in order to exclude low read content barcodes, which we've determined primarily correspond to just ambient DNA. And so by doing this thresholding, we then enrich for real cells, essentially. And next, we can look at the distribution of these cells across both this 96-well and isolate chip, where you can see here we're capturing slightly higher number of cells for this repressive modification, likely because we're also capturing a larger library per cell. And then here in this particular experiment, we actually performed a serial dilution across the source wells of the iCell8, where we do a 1-to-2 dilution, which is then dispensed 2 times across the top half of the chip and the second half. And you can see that indeed, on average, we are capturing the number of cells we expected to dispense. So again, that's a good sign. And next, we just took a global view of the aggregate signal of both modifications. And what you can see just on this zoomed out view is that indeed our repressive chromatin modification and active modification, as we would expect, are labeling distinct regions of the genome. And you may be here we're just showing a finer scale of that, a zoomed in version over the Hoxsay cluster. where again we see, as expected, that H3K27 trimethylation forms these broad repressive domains, whereas the H3K4 methylation forms these more peaky signal over transcriptionally active promoters and distal regulatory elements such as enhancers. And that difference in the peak size is just quantified here. And then we just compared the fraction of reads that fell in peaks, where we can see that indeed H3K27 trimethylation is much more enriched in the repressive domains than these active peaks, and the opposite is true, as we would expect, for the histone H3 lysine 4 methylation mark. So now we're ready to actually begin to do the QC at a single cell resolution, and this is where mixing the two different donors becomes important. And so, as with any single cell platform, one thing that we want to, first quality control that we want to do is identify barcodes that correspond to actually collisions between multiple cells and aren't actually representative of true single cell data. And so by mixing multiple donors, we can take advantage of these SNP-based doublet calling tools. And for this particular study that I'm showing you, we used a tool called Supercell, cited here. And essentially, this program doesn't require any a priori information as to the genotype of the donors, but rather uses the single cell data as input in order to identify SNPs that are associated with one another at a much higher than would be expected by chance, and likely correspond to SNPs within an individual donor, and can then identify barcodes that contains combinations of SNPs that it had assigned to distinct donors and label these as doublets or collisions, which we've shown here as purple in these UMAPs for both the repressive mark here on top and the trend and the active modification on the bottom. And when we compare this to other doublet calling methods that use synthetic doublet strategies, such as the method that's baked into the ArchR platform, you can see that this compares favorably, and they both identify a overlapping set of doublets. But we really like the SNP-based method because it's an orthogonal strategy that really provides a ground truth of which cells are most likely corresponding to collisions. And we can then use this doublet information to go through it several rounds of proximity-based embedding and throw out clusters that are enriched for doublets to clean up the data. And this was done in collaboration with a talented graduate student in the lab named Jacob Green, where you can see he was able to clean up these UMAPs and resolve clusters that, based on the signal of either the repressive modification or active modification, could be annotated as each of the various cell types which are expected within peripheral blood mononuclear cells. And next, Jacob went on to basically confirm these annotations by taking bulk ChIP-seq data that had been generated as part of the ENCODE project and projecting it onto our UMAP. So again, you can see here that these bulk data largely projects into the expected clusters, confirming that this method was able to identify each of these cell types based on both the repressive chromatin regions and active chromatin regions. So in summary, what I've shown you is that CUT&Tag facilitates the genomic profiling of chromatin-associated proteins at single-cell resolution, and that Sky CUT&Tag with a nanowell dispenser such as the ISL8 can dramatically increase the number of high-quality single-cell profiles captured in a single-cell experiment, such that now, in a given experiment, we're capturing between 30 and 100,000 single-cell profiles. And we can then use this strategy of SNP-based doublet removal to clean up the SCI cut-and-tag data in silico. And so last, I just want to tell you about another way or modification to this method that I worked out in collaboration with another postdoc in the lab, Michael Mears, who's now gone on to start his own lab at WashU, in which we modified the SCI cut-and-tag approach for multifactorial profiling, where we're able to profile multiple chromatin-associated proteins within individual cells. And to do this, we basically performed iterative rounds of antibody labeling and tagmentation with differentially barcoded adapters, such that each chromatin-associated protein is now labeled by differentially barcoded adapters, such that based on those barcodes, we can then demultiplex the signal and determine which protein they correspond to. So again, this then gets dispensed across an iCell 8. And here I'm just showing you a snapshot of the aggregate data performed on top in bulk, and then the aggregate single cell data, which you can see corresponds nicely here on the bottom. And the power of this approach, as I was mentioning, is it now allows us to match up profiles for different proteins within individual cells and look at how, say, the repressive genome and active genome may be changing in concert with one another. And so that, in conclusion there, this multi-tag is an efficient, cost-effective method to profile multiple chromatin modifications in individual cells. And so with that, I'd just like to acknowledge everyone in the Henikoff lab, in particular, Steve, for his guidance throughout this project, as well as Mike, who helped me with the multi-tag approach, as well as Jacob Green, who joined the lab after this retreat, which is pictured here. And with that, I'll take any questions you may have. Thank you very much for your time.
Thank you, Dr. Janssens, for your presentation. I will now pass this over to our second speaker, Dr. Iacobucci. You may now begin your presentation.
Hello to everyone. I would like to thank Takara for the kind invitation and the opportunity to share the results from the application of single-cell sequencing approaches to the study of acute leukemia. My research is focused on high-risk leukemias, including both acute myeloid leukemia and acute lymphoblastic leukemia, and the overall goal is to comprehensively define the genomic basis of these diseases by high-throughput profiling of genome, transcriptome, and epigenome at both bulk and at the single-cell level. And once the genetic alterations are identified, the aim is to develop faithful experimental models that recapitulate human leukemia and provide a platform for exploring not only the functional role of these alterations, but also the potential benefit of novel therapies. In this presentation, I will provide 2 examples of how the application of single-cell sequencing has enabled the dissection of transcriptomic heterogeneity in models of acute erythroid leukemia, and has allowed it more recently an accurate reconstruction of clonal evolution by high-throughput single-cell whole genome sequencing in a subset of B-cell acute lymphoblastic leukemia with intrachromosomal amplification of chromosome 21. So starting from the first example, I would like to provide a little bit of background about acute erythroid leukemia, which is an high-risk leukemia subtype characterized by uncontrolled expansion of erythroid progenitor cells, with still many controversies in disease classification and continuous changes in classification criteria. These changes definitely will impact clinical management and treatment options for this disease. This is a poor prognostic form of leukemia with dismal outcome to conventional chemotherapeutic agents. Given the lack of non-genomic features and the dismal outcome, a few years ago we started an international study aimed to collect as many samples as possible worldwide to understand the genetic basis of this disease. Overall, we collected and analyzed by genomic analysis 159 cases, including RNA-seq, exome sequencing, and whole genome sequencing. And we compared the data from these patients to those from almost 2,000 non-erythroid acute myeloid leukemia and myelodysplastic syndrome cases. Although morphological criteria alone have failed to provide meaningful insights into leukemic biology and classification, here we showed that acute erythroid leukemia is characterized by a distinct mutational spectrum different from other myeloid, non-erythroid, AML, or MDS subtypes, with 6 age-related genomic subgroups having distinct gene expression profile. TP53 mutations, often with concomitant mutations of chromatin regulators, transcription factors, or tumor suppressors, are the most frequent alterations, occurring in over 32% of cases and in almost 50% if considering only the adult cases. These mutations are associated with a very poor prognosis. Since experimental models of acute erythroid leukemia are limited or did not recapitulate the complexity of this disease with often more than one mutation present per sample, we aimed to establish functional experimental models to better understand the role of mutations into leukemogenesis and to explore the potential benefit of novel targeted therapies. We took advantage of CRISPR-Cas9 genome editing to induce combinatorial mutations in 10 genes recurrently mutated in acute erythroid leukemia and affecting key pathways such as the regulation of apoptosis and cell cycle, DNA methylation, chromatin, and transcriptional regulation. And according to the pattern of cooperation and mutual exclusivity of mutations in patients, multiple lentiviral pools with different combinations of single-guide RNA against these genes were generated. This is a cartoon showing the workflow of this study. So these pools were used to induce multiplex genome editing in Cas9, GFP, knock-in mouse, hematopoietic stem, and progenitor cells that were used subsequently in transplant assays. Established tumors were analyzed by a plethora of studies, including single-cell DNA and RNA sequencing, to dissect the genetic heterogeneity induced by genome editing at the single-cell resolution. Cross-species gene expression analysis to investigate if these models recapitulated human leukemia. Moreover, these tumors provided a powerful platform for exploring therapeutic intervention. The first results that I would like to highlight in this presentation is that in contrast to the uniform representation of single-guide RNA in hematopoietic stem and progenitor cells pre-transplant, developed tumors showed an enrichment of specific guides RNA with a phenotype that was directly related to the enrichment of these guides RNA. For example, concomitant editing and inactivation of TP53 VCAR DNMT3A promoted the development of acute erythroid leukemia as demonstrated by gene expression, immunophenotype, and immunohistochemistry. Other combination of mutations induced a B or a T phenotype or a mixed lymphoid erythroid phenotype, where I will go back later in this presentation. Whole transcriptome sequencing revealed specific subgroups associated with the leukemia lineage, and we observed the enrichment of transcription factors required for differentiation of megakaryocyte erythroid progenitor cells, confirming the erythroid feature of these models. Moreover, by cross-species analysis, these data showed signatures similar to human erythroid leukemia with similar up and down regulated genes, confirming that these models faithfully recapitulated human leukemia. Interestingly, in one tumor, we observed the phenotypic evolution across serial transplant, and specifically in a model with the mutations in BCAR, TP53, and RB1. We observed the expansion of cells with the mixed acute erythroid leukemia, a B-cell lineage phenotype in secondary passages. So as shown in this slide by immunohistochemistry, these tumors had both myeloid, lymphoid, and erythroid markers on the same cells. To investigate further phenotypic and transcriptomic changes associated with the lineage clonal evolution, we performed single-cell full-length RNA sequencing by the full-length SMART-seq chemistry and smarter isolate single-cell system. For this approach, cryopreserved mouse leukemic cells were thawed, stained with ox and propidium iodide, dispensed into a microchip, and imaged for selection of viable and single cells, and then processed for single-cell DNA synthesis, amplification, indexing, and the library preparation for sequencing. We sequenced around 500 cells per each sample at a coverage of 1 million reads per cell, and the reads were analyzed for gene quantification and analysis of different expression groups. We identified an average of 2,000 expressed genes per cell, but I would like also to highlight that this was the first study that we did with this chemistry, but lately we are using a new chemistry from SMART-seq PROC that allows us to reach over 10,000 expressed genes per cell, improving the detection of splicing alternative isoforms and gene fusions in leukemic cells. By SRAT analysis, we identified 4 distinct gene expression clusters. These are t-SNP plots in which each cell is a dot, and in the first image on the left, cells are colored by transplant passage with the primary tumor in red and secondary tumor in blue, while in the right panel, cells are colored by gene expression clusters. So we noticed that more immature hematopoietic cells with the stemness phenotype and expression of both erythroid and lymphoid primitive markers and progenitor erythroid cells expressing GATA1 were dominant in the primary tumor, while more differentiated erythroid cells expressing GATA1 and CD71 and more differentiated mixed B-cell lymphoid and erythroid cells were dominant in the secondary passage. The secondary tumor showed upregulation of B-cell receptor signaling and germinal center B-cell signatures with expression of genes driven by BCL6. Interestingly, bulk sequencing showed for the secondary tumor the same mutations present in the primary tumors. However, only by full-length single-cell RNA-seq, we were able to identify an alternative isoform of BCAR only in this mixed B erythroid population, and this isoform was due to a larger BCAR deletion. This deletion was further confirmed by single-cell DNA sequencing of total and purified subpopulations of erythroid, lymphoid, or mixed phenotype, confirming that this deletion was only restricted to cells with the mixed erythroid, lymphoid phenotype. BCAR is a component of the non-canonical polycomb repressive complex 1, which is involved in many biological processes, including maintenance of stem cell identity, differentiation, and cancer development, and in various cellular functions, including post-translational histone modification and chromatin compaction, and thus it provides a link between transcriptional and epigenetic regulations. Moreover, mutations of BCAR occur across different hematologic malignancies. So in contrast to the truncated BCOR alleles induced by genome editing in the primary tumor, expression of this shorter BCOR isoform was associated with the downregulation of components of non-canonical polycomb repressive complex 1 and with the overexpression of its repressed target genes, including lymphoid markers as shown here in this slide. The interaction region with BCL6 was retained as demonstrated by upregulation of BCL6 and B-cell receptor signaling pathway, suggesting that this BCOR isoform may promote lymphoid maturation in these cells. So overall, these data demonstrated the ability of single-cell transcriptome sequencing to successfully characterize different leukemic subpopulations, and in this case allowed us to demonstrate a phenotypic evolution in mouse serial transplant. The second example that I would like to highlight in this presentation is represented by the reconstruction of genetic clonal evolution by high-throughput single-cell whole genome sequencing in a subtype of B-cell acute lymphoblastic leukemia with intrachromosomal amplification of chromosome 21. This subtype includes up to 5% of all BLL cases. TCU map in this slide shows the different subtypes of BLL. There are over 23 different subtypes here colored by the driver genetic alterations, and each dot is a sample. So you can see that these subtypes are characterized by distinct gene expression profiles. The subtype here in purple with the intrachromosomal amplification of chromosome 21, also shortly defined IM21, is characterized by gains occurring on chromosome 21 and complex rearrangements comprising multiple regions of inversion, deletion, so not only gains. And here, this picture highlights all the changes within red copy number gains and in blue copy number losses on chromosome 21. In most cases, the formation of the IM21 chromosome involves one or more breakage fusion bridge cycles followed by chromotripsis of chromosome 21, so complex genetic chromosomal rearrangements. To get insights into clonal evolution of cells with this type of alteration, we performed multimodal single-cell sequencing, including single-cell RNA-seq and single-cell whole genome sequencing. First, from single-cell RNA-seq, we inferred CMV, and we observed that there are 3 different copy number clusters. Almost all cells show amplification of chromosome 21, meaning that this alteration is one of the earlier alterations occurring in this type of leukemia. But we also observed 2 smaller clones, one that had chromosome 21 only, and not the secondary alteration that was observed in the predominant clone and involved the amplification of chromosome 14. And then a smaller clone here in gold with extra gain of chromosome 21, but lack of a gain of chromosome 14. So to validate this inferred observation of clonal heterogeneity, we performed single-cell whole genome sequencing of flow-sorted blast and normal cells in order to explore better the copy number states of chromosome 21 and chromosome 14 across different cells. For this approach, frozen mononuclear cells were thawed, stained with antibodies for normal and leukemic populations, single cells sorted in 96 well plates and processed by the Pacoplex from Takara. This workflow consists of 4 main steps, including lysing, extracting, and amplifying the genomic DNA from single cells, and then preparing a library prep for Illumina sequencing. Bulk whole genome sequencing data identified on chromosome 21 9 segments with very copy number patterns represented here by different regions. However, bulk whole genome sequencing failed to detect the clonal heterogeneity to provide insights on clonal evolution. In contrast, the single-cell whole genome sequencing confirmed the InfluoCMV from single-cell RNA-seq and allowed us to dissect the clonal heterogeneity for chromosome 21 copy number changes. Data are summarized in this scatter plot in which each column is a cell, and for each genomic segment identified by bulk whole genome sequencing, the copy number status by single-cell whole genome sequencing is reported with in red gains and in blue losses. So these cells are assigned to different clusters labeled as C1, C2, and C3 based on their copy number patterns. So again, these results show that there is a predominant clone with different gains of chromosome 21 and amplification of chromosome 14, a smaller clone with only gain of chromosome 21, and a smallest clone with extra gains of chromosome 21. And so overall, these results allowed us to build a model in which gain of the IM21 chromosome occurs earlier than gain of chromosome 14, and the IM21 chromosome undergoes progression amplification, further increasing the expression of genes in the common region of the amplification. And so the cartoon here depicts this model with a single clone at the beginning with amplification of chromosome 21, and then two clones, one that acquires chromosome 14 and the others that acquires extra gains of chromosome 21 but lacking chromosome 14. By the same approach, we analyze a second sample with different copy number alterations involving this time more chromosomes, such as chromosome 1, 11, 21, and X, as shown by the bulk whole genome sequencing. And again, by single-cell whole genome sequencing, we found three clusters. Cluster 1 with the IM21, loss of chromosome 1q, loss of 11q, and gain of chromosome XQ. Cluster 2 with the IM21 and the gain of chromosome XP, and another cluster with IM21, gain of chromosome XP and loss of 1q. So again, this data enforce the model that chromosome 21 gain occurs earlier of other chromosomal alterations, including in this case, loss of chromosome 1, 11, and gain of chromosome X. So in conclusion, this analysis by whole genome sequencing allowed us to build a model of clonal evolution in which acquisition of the IM21 chromosome is an early event, potentially followed by progressive amplification of regions on chromosome 21, increasing the expression of genes included in this region. And single-cell whole genome sequencing by the Picoplex platform enabled us to detect small clones with different copy number states. Finally, I would like to thank Charles Mulligan Laboratory, Shin-Sung Gao, and all the collaborators that contributed to some of the data I presented. And thank you all for attending this webinar.
Thank you, Dr. Janssens and Dr. Iacobucci, for your informative presentations. We will now start the live Q&A portion of this webinar. If you have a question you'd like to ask, please do so now. Just click on the Ask a Question box located on the far left of your screen, and we'll answer as many of your questions as we have time for. Let's get started. Our first question is for Dr. Janssens. How many nuclei do you recommend to start with for an iCell8 experiment? Does this amount differ between the protocol that uses Con-A beads versus the one that uses centrifugation for the wash steps?
Hey, yeah, that's a great question. So for a typical iCell8 experiment, I mean, if you're not doing the combinatorial indexing method, you can start with, very few cells. if they're bound to Kane beads, they're retained well and can be dispensed. And so you could start with as few as, say, a few thousand. But then again, we'll usually capture, say, 1,000 cells or 1,000 to 2,000 cells per experiment. For the combinatorial indexing strategy, there's not really a lower limit, but we do set an upper limit of 2 million. And that's just because basically once you go above that, cell aggregation actually becomes more of a problem. So clumping. And I think the reason is that the aggregation is primarily driven by lysis. And so as long as the number of cells is kept below 2 million, that lysis is limited enough that there's enough cells that are retained in a single cell suspension to dispense, 10 to 20 cells per nanowell. And then in terms of how does this amount differ between a protocol that uses Kane beads versus one that uses centrifugation for wash steps, I think for doing centrifugation during wash steps, cellular pelleting basically is more efficient with more cells. So having more material is probably more critical for doing the centrifugation. But generally in the lab, I think we've moved away from doing that strategy.
Wonderful, thank you. It looks like our next question is also for you, Dr. Janssens. How do you prevent clumping of Kane beads after tagmentation, or overall, what can you do to minimize nuclei clumping throughout the protocol? Can you put the nuclei through a cell strainer when using Kane beads before dispensing nuclei on the iCell8?
Yeah, so one of the early steps in the protocol, and actually during nuclei preparation, is we do a light cross-linking step. So that's using 0.1% formaldehyde for 2 minutes and then quenching it. And that cross-linking, I think, because the main driver of aggregation is lysis, I think that cross-linking serves to basically help hold the nuclei intact and prevent lysis throughout the protocol. But of course, some amount of lysis still occurs. And so as you suggested in this question, yes, we always essentially after doing the tagmentation, the 96 well plate, and then recombining everything into a single tube, we then pass the whole sample through a 20 micron strainer, which effectively removes aggregation. And I should also state, for people who are interested in this combinatorial indexing method or just doing single cell cut and tag in general, we've also been working to develop a whole cell protocol. And that seems to work very effectively as well, where we just permeabilize whole cells and lightly cross-link them and then run essentially the same protocol. And so that's helped to streamline the workflow a little bit, even since this initial nuclei protocol was developed.
Fantastic. Looks like our next question here is for Dr. Iacobucci. Why did you choose SmartSeq Pro over 10x or droplet-based end counting methods?
Thank you for this question. The main advantage of using SmartSeq Pro is the ability to capture full-length transcriptome. We know that in leukemia and other tumor subtypes, there is a lot of genetic heterogeneity with gene fusions, mutations, and the ability to capture those rearrangements as well as splicing isoforms is important for correlation with subsequent gene expression profile. And as we were very interested to correlate genetic alterations such as fusions to their own gene expression profile. And this is possible by the full-length chemistry of the SmartSeq Pro. Moreover, another advantage of the Pro chemistry is the ability to detect over 10,000 expressed genes. With the other approaches, we are below that number. And so with the higher number of genes, we have more reliable clusters done in the gene expression analysis.
Wonderful, thank you. And another one for you. What sequencing depth did you usually use for the SmartSec Pro libraries?
Right. So since we have a full-length transcriptome and not only like the end of the transcript, so we need to go higher with coverage compared to other like approaches. And so we do at least 500,000 reads per cell. The optimal that we do is 1 million reads per cell.
Thank you. And it looks like we have some more questions here coming in for you. And go ahead and submit any questions in the Ask a Question box, lower left corner of the screen. We'll answer some more questions here before we wrap up. But our next question for you, Dr. Iacobucci, is what kind of cell population in the B-cell lymphoma model did you use for scRNA-seq? How many cells, fresh or frozen cells?
Okay, so in the mouse models, that was not only B cell, but was a mixed erythroid B cell. So we used around 500 cells. There was a frozen sample since these were tissues that were harvested from mice that developed tumors. And so we cryopreserved tissues. It did a plethora of analysis, including bulk sequencing. And then in order to investigate better the genetic heterogeneity, we went to single cell. And so we started with like 500 because in the same chip for the isolate, we combined two different samples. And so we had an average of 500 cells. We also, like thereafter, did some experiments where we compared the fresh versus frozen cell lines, always for the isolate. And we honestly did not observe any differences in terms of data and clustering. However, I would like to mention that to enrich the viable cells, before we go on the isolate platform, we perform that cell removal. So even if we stain with propidium iodide before going to the isolate, we always do that cell removal in order to enrich and make sure that we have as many cells as possible to be captured.
Fantastic, thank you. And it looks like a follow-up question for that maybe. Except scRNA-seq, what other approaches did you use to identify the BAL model?
Yeah, as mentioned, we use a lot of approaches because these models were established by CRISPR of many different genes that were used in a pool. And so in order to identify the genetic alterations that were induced by genome editing and the tumor that the phenotypic feature of the tumor that was established, we use conventional approaches. So we started from flow cytometry, including several markers for the main hematopoietic lineages. We did immunohistochemistry, especially because for acute erythroid leukemia, some of the key transcription factors may not be detected by flow cytometry. And we did bulk RNA-seq, bulk whole exome sequencing, and then we did targeted the next generation sequencing to identify the specific genome editing induced mutations. And then we did single cells sequencing, both for targeted DNA and for RNA-seq. And so in this way, we could compare the genetic variegation with the phenotypic feature of those models.
Fantastic. Thank you so much. It looks like that's all the questions that we have time for right now, but I did want to take this opportunity to thank both Dr. Janssens and Dr. Iacobucci for your time today and your important research. We would also like to thank LabRoots and our sponsor, Takara Bio, for underwriting today's webcast. Before we go, I wanted to ask Dr. Janssens, did you have any final comments for our audience?
Yeah, thanks a lot for attending. And the one thing I guess I would just like to mention is that if you're interested in using any of the methods that were developed in the Henikoff lab, cut and run or cut and tag, or what I described here, single cell cut and tag, we maintain a number of protocols.io websites or pages that have active discussion forums, and you can post any questions there. And we usually respond to those very quickly. And also the protocol that I described today for combinatorial indexing using isolate is currently under review, so we expect that will get published shortly. So keep an eye out for that one. Thanks.
Thank you. And Dr. Iacobucci, did you have any final comments today for our audience?
Yes, first of all, I also would like to thank the audience for attending our presentation. It's been an honor for me to present our data. And also, if you have any questions regarding the single-cell RNA-seq, the single-cell whole genome sequencing, please do not hesitate to shoot me an email. And also, I'm always very interested in testing new approaches, and so I'm also flexible in that, in testing new assays on the isolate as well. So any inputs is appreciated.
Fantastic. Before we go, I'd like to thank the audience for joining us today and for their interesting questions. Questions that we did not have time for today and those submitted during the on-demand period will be addressed by our speakers via the contact information you provided at the time of registration. This webcast can be viewed on demand. LabRoots will alert you via email when it's available for replay. We encourage you to share that email with your colleagues who may have missed today's live event. Until next time, goodbye.